So, kind of gave an overview, therapeutic window, side effects, lethal dose, effective dose, and now let's talk about the actual development of a drug. And what is the process to bring a new medicine to market? Well, it consists of a total of 5 phases. First phase, and this is our overview, we'll talk a little bit more about the actual development process in detail. Phase 0 is that drug discovery and preclinical trials. So you're trying to synthesize a new drug, or you're trying to isolate it from a existing person-animal substance. Phases 1 through 3 consist of those clinical trials, and those are followed by the FDA approval. And then phase 4 is once you've got it to market and you've covered the basics for safety, it's post-market monitoring. You're opening it up to a larger population, and you want to make sure that the side effects are still tolerable and that the effectiveness of the drug is still within acceptable parameters. So the drug discovery process, this can be up to 12 years as a process. So 12 years for a company, that is a big time and resource commitment by these companies to develop these drugs and bring it to market. Gosh, commitment. Okay, so that's a big time and resource commitment by companies to bring these. Okay. So we have the discovery phase. Okay, that's identification of lead compounds, not lead, it's lead compounds. Synthesis of analogs, biological testing, and we start off with about 5,000 plus drugs. Okay. And that discovery process lasts about 3 years. At the end of 3 years from that 5,000, we've got 8 to 15 viable drugs to take to the next phase. This is the development and research phase, your phase 1, phase 2, phase 3, and you whittle that 8 to 15 down to 1. Then you go into the regulatory review and then that post-market monitoring. Taking a little closer look at this phase 0. Again, this is lead, not lead, lead. Okay. So in phase 0, we're going to identify compounds that have biological activity. We're going to screen chemically related compounds. These are called analogs. And these methods include combinatorial chemistry. And this is where we can synthesize compounds that are related to the lead compound, and we do that in a high throughput way, meaning many compounds in a short time frame. So you're making a lot and screening them very quickly. And then this is somewhat recently developed, and this is to speed up that drug discovery process. You have to develop a manufacturing process. Okay, you want to make sure that what you are making, or I should say what you are testing, you are capable of making in marketable quantities. The idea is to make a profit when these compounds come to market. Okay. All right, and then preclinical testing. Preclinical testing is, we don't really need to copy this. Okay, this is kind of the overview. Okay, so you get a pharmacological or manufacturing profile of your selected compound. You're going to evaluate that compound's safety, potential toxicity in animal models first. You're going to determine whether it's toxic Carcinogenic, meaning is it a cancer-causing compound? Mutagenic, is it going to mutate genes? Pterogenic, pterogenic. Can the body absorb the drug? How is it metabolized? What are the metabolites? What are their effects? How long does it remain in the bloodstream? And where does it go? Okay. Also happening in this preclinical testing, formulation and manufacturing processes to be used for testing with humans. Specifications to evaluate the chemical quality. You're going to be looking at the purity, the stability, the reproducibility of the manufacturing process. Preclinical studies also help researchers design the human phase 1 studies. What initial dose will it be started at? What symptoms are we going to be looking for? And a IND, an investigational new drug application, is filed with the FDA. If that gets approved, then clinical trials can begin, and that really means start testing on people. Looking at phase 1 clinical trials, and we're really focusing on this phase 1 in safety. This is going to be a first time that a drug is tested in humans. Really just making sure that it is safe and that there are no big issues. Okay, so in this first phase, there's a small number of healthy volunteers, or they may include patients with specific conditions for which the targeted pathways have been identified as potentially relevant to the disease that's under study. This phase can last several months. And what the companies do is they focus on the safety, the dosage range, the idea of any side effects, and the detection of early evidence of the effectiveness. Phase 2 are the clinical trials, and this is proof of concept. The drug is tested for the first time in patients that have the disease or the condition. Several hundred patients are tested, and this can last several months to a few years. The dosages are adjusted, side effects again are determined, and the best regimen to take it to larger clinical trials. In this initial proof of concept, okay, you're really proving the drug did what it was intended to do. Did it interact correctly with its molecular target? Did it alter the disease? And then phase 2B, larger number of participants. You may use that to compare to other drugs. And then there is a larger, broader dosages to obtain a much more robust proof of concept. The phase 3 of the clinical trials, and the one thing that we need to insert here, sorry about this, is, and this is an important thing, and you do need to know this, is the placebo effect. A placebo is essentially a sugar pill. Okay? And within these clinical trials, people are given sugar pills to test a psychological factor. All right, so in these trials, a patient is given a sugar pill. Okay, and this really is pertaining to the phase 3 clinical trials. It's given a sugar pill. Okay, they think it is the drug. When this happens, sometimes there is a psychological effect where the patient thinks they're getting better. Okay. In some cases, they do get better. You know, the power of positive thinking. If you think you're getting a drug to cure something, you actually feel better, sometimes, I guess, just over a course of time, you get better, right? And that's known as the placebo effect. And again, the placebo effect is really in these phase 3 trials. Okay, so phase 3 clinical trials, these are required for the FDA approval of the drug. These can take place over several years, multiple clinical centers around the world, several hundred to several thousand patients. The purpose of these trials is to provide the proof needed to satisfy regulators that the medicine meets the legal requirements to be approved for marketing. There is a double-blind study. Neither the patient nor the researcher knows which patients are receiving the drug and which patients are receiving placebos during the course of this trial. It is important that neither the patient nor the researcher, because there can be bias on either end. That's why it's called double-blind. If these phase 3 trials are successful, then an NDA is submitted to the FDA, and this review usually takes 10 to 12 months. And that's this part right in here. This regulatory is usually like 12 months. And then we have the phase 4 of the clinical trials. This is after approval. You still need to follow the drug. Okay. And this is to make sure that it still continues to do what it was designed to do. So in that phase 4, there's medical marketing. Studies conducted after the drug receives the regulatory approval. The FDA may require companies may voluntarily undertake a post-approval studies to generate additional information about the drug. Post-approval trials also are designed to test the drug with additional patient populations. Maybe you expand into pediatrics. If there's any new delivery modes, time-release capsule versus just a regular pill. If there are any new uses or indications. Okay, sometimes it'll be effective for a different medical condition than the one that it was intended for. These phase 4 have to meet the same standard as the phase 3 trials conducted for initial approval. All right, and some examples of drugs that had some post-market problems. It would be thalidomide, which causes birth defects, and Vioxx, which was used as an anti-inflammatory and then later found to cause heart attacks. Oh, look. I didn't know how to slide. Sorry about that. All right, so thalidomide was given to pregnant women to stop miscarriages and also to help with morning sickness, was my belief. And it caused birth defects, and these were mainly limb abnormalities. And then Vioxx, anti-inflammatory, but it was shown to cause heart attacks and strokes. Now I think it's the end.